Hello everyone, welcome to the Concept and Fact of Life Sciences. Today in this video, we are going to discuss about the cell organelle which occupies maximum space in the cell, that is the endoplasmic reticulum. The endoplasmic reticulum, it is a single membrane bound cell organelle. The cell organelle on the basis of number of membranes they have can be categorized into three categories, that is the double membranous cell organelles, which includes the nucleus, mitochondria and plastid. There is a non-membranous cell organelle in the form of ribosome and proteasome, and there is a presence of some single membranous cell organelles in the form of endoplasmic reticulum, Golgi body, etc. So the endoplasmic reticulum, it falls within the category of cell organelles that remain surrounded by a single membrane. Now this endoplasmic reticulum, it is generally composed of three things. There is a presence of flattened plate-like structure which are known as cisternae. There is a presence of tubule and the vesicles. So it is generally composed of cisternae, tubules and vesicles. Now the endoplasmic reticulum, they are mainly of two types. There is a rough endoplasmic reticulum and there is a smooth endoplasmic reticulum. Now if we talk about the rough endoplasmic reticulum, these are the plates where the ribosome binds. So the major function of rough endoplasmic reticulum is the protein synthesis. For this, there is a binding of ribosomes. So generally we can say that these are the plates where there is a binding of ribosomes. Now the ribosome binds to these plates only when the cell is synthesizing the protein. जब सेल प्रोटीन का सिंथेसाइजेशन कर रहा है तभी राइबोसोम्स जो हैं रफ एंडोप्लाज्मिक रेटिकुलम से बाइंड होते हैं. So it means कि if the cell is not synthesizing the protein, then the rough endoplasmic reticulum is equivalent to the smooth endoplasmic reticulum? No. They are still different from the smooth endoplasmic reticulum because they contains the proteins which are known as ribophorines. There is a presence of some proteins on the membrane of rough endoplasmic reticulum and these proteins they are called as ribophorines. Ribophorines they are generally transmembrane glycoproteins. ये होते हैं आपके transmembrane glycoproteins. These are the proteins which recognizes or which are recognized by the ribosome for their attachments, we can say. So they help in the attachment of ribosomes on the plates or on the rough endoplasmic reticulum. So still, if the cell is not synthesizing the protein, the rough endoplasmic reticulum are different from the smooth endoplasmic reticulum in having the protein which are known as ribophorines, and these ribophorines, these are the transmembrane glycoproteins. Now in case of rough endoplasmic reticulum, the cisternaes are well developed. Now these flattened plates, these are called as the cisternae. As we know that the major function of rough endoplasmic reticulum is the protein synthesis. So it generally wants to maximize that particular area which we can say involved in the process of protein synthesis or where the ribosomes they are binding. That's why in case of a rough endoplasmic reticulum, these cisternaes they are well developed to provide maximum space for the binding of ribosomes. Now if we talk about the smooth endoplasmic reticulum, in contrast to the rough endoplasmic reticulum, the major function of smooth endoplasmic reticulum is the carbohydrate metabolism and lipid biosynthesis. Now there is a synthesization of all glycerol phospholipids within the smooth endoplasmic reticulum, or we can say there is synthesization of all phospholipid except the sphingolipids. जैसा कि अपन ने बात की थी, जब अपन ने बात की थी प्लाज्मा मेम्ब्रेन के स्ट्रक्चर के बारे में, there are different types of lipids, or we can say lipids they can be categorized, plasma membrane lipid can be categorized into two categories on the basis of their framework alcohol. One is the glycerol and other is the sphingosine. So the glycerol derivatives or the glycerol phospholipids they can be synthesized within the smooth endoplasmic reticulum, while the sphingolipids they synthesizes within the Golgi complex, जिसके बारे में अपन आगे बात करेंगे. So there is a synthesization of glycerol phospholipid within the smooth endoplasmic reticulum. Next function of smooth endoplasmic reticulum is the detoxification. So this smooth endoplasmic reticulum, they are also involved in the process of detoxification. They contains an enzyme which is known as cytochrome P450 monooxygenase that converts the active drugs into the inactive drugs. कैसे करेगा उसके बारे में आगे बात करेंगे. In case of the smooth endoplasmic reticulum, because they are mainly involved in the process of secretion, so there are tubules which are well developed in case of smooth endoplasmic reticulum. जो rough endoplasmic reticulum है, the cisternes are well developed, while in case of the smooth endoplasmic reticulum, the tubules are well developed. So these are some basic difference between the rough and the smooth endoplasmic reticulum. एक एक करके इन दोनों के बारे में बात करते हैं. एक चीज और यहाँ रह गई है, जैसे कि अपने पहले भी बात की थी, द आउटर मेम्ब्रेन ऑफ द न्यूक्लियस, दिस द आउटर मेम्ब्रेन ऑफ द न्यूक्लियस, इट विल एक्ट एज द मेम्ब्रेन ऑफ एंडोप्लाज्मिक रेटिकुलम. ठीक है, कुछ बुक्स के अंदर ऐसा कहा गया है कि एंडोप्लाज्मिक रेटिकुलम इट इज ए डबल मेम्ब्रेन बाउंड सेल ऑर्गेनल, बट इट इज ए सिंगल मेम्ब्रेन बाउंड सेल ऑर्गेनल, एंड द आउटर मेम्ब्रेन ऑफ द न्यूक्लियस इट विल एक्ट एज द मेम्ब्रेन फॉर एंडोप्लाज्मिक रेटिकुलम. बात करते हैं पहले रफ एंडोप्लाज्मिक रेटिकुलम के बारे में. सो if we talk about the functions of rough endoplasmic reticulum, the first and the major function of rough endoplasmic reticulum is the synthesization of proteins. Now the protein on the basis of their site of synthesis, जैसा कि अपन ने बात की थी in our previous video on protein sorting and targeting, the protein on the basis of their site of synthesis, they are of two types. One is secretory, other is known as the cytosolic protein. Cytosolic proteins, their synthesis starts within the cytosol. The translation process, it completes within the cytosol, and they functions within very many cell organelles like the mitochondria, nucleus, and the chloroplast. While the other category of protein, it is the secretory protein that synthesizes within the lumen of rough endoplasmic reticulum. कैसे होता है वो भी अपन पहले डिस्कस किया था, थोड़ा सा प्रोसेस वापस समझ लेते हैं. We have nucleus. Within the nucleus we have DNA. DNA will convert into the mRNA through the process of transcription. Now then the mRNA, it comes into the cytoplasm through the nuclear pore complex. Now we have mRNA within the cytosol. Within the cytosol there is a presence of free ribosomes. These ribosomes, they attaches to the mRNA, and then the process of translation it starts. The initial polypeptide or the initial amino acids which are synthesized during this process, if they contains the signal peptides, or if there is a presence of some hydrophobic amino acid that constitutes the signal peptides. These signal peptides, these are the signals for the transcript, or we can say these are the signal for this protein to be transported into the lumen of endoplasmic reticulum. For this transportation, we have a adapter molecule which is known as SRP. It is basically a ribonucleoprotein which is also known as small cytoplasmic RNA. It recognizes the signal peptide, and after the recognition of we can say signal peptide by the SRP, that is the signal recognition particle, this SRP brings this complete transcript, we can say brings this complete transcript onto the membrane of rough endoplasmic reticulum. Because the rough endoplasmic reticulum membrane, it contains the SRP receptor that can recognize or that can interact with the SRP. So there is an interaction of SRP with the SRP receptor. Now this SRP receptor, it is made up of two subunits. One is alpha, which means present toward the cytosol, and other is known as the beta, which is a transmembrane domain. Both of these subunits, they contains the GTPase activity. Now the interaction of SRP with the alpha subunit of the SRP receptor, it activates the GTPase activity of alpha subunit. The GTP binds to the alpha subunit. There is a conformational change in alpha which makes conformational change in the SRP. Then this SRP, it will be released into the cytoplasm for next round of translocation. Now after this, the alpha and beta subunit, they also recruits the SEC61 class of transporters for the transportation of newly synthesized polypeptide into the lumen of endoplasmic reticulum. Within the lumen of endoplasmic reticulum, we have a protein enzyme which is known as signal peptidase. Now, this signal peptidase, the function of this signal peptidase is just to cleave, you can say just to cleave the signal peptide. Now, after the cleavage of signal peptide by the enzyme signal peptidase, there is a complete synthesization of polypeptide within the lumen of endoplasmic reticulum. So, we can say that the major function of rough endoplasmic reticulum is the synthesization of secretory protein. Now, beside this, they have very many functions, जिसके बारे में अपन बात करते हैं. Within the lumen of endoplasmic reticulum, we have a BIP protein. Basically, it is a binding immunoglobulin protein which belongs to the category of HSP 70 class of chaperone. Now this, the function of this BIP protein, बात कर सकते हैं अपने यहाँ पे राइट डाउन भी किया है. Binding immunoglobulin protein, it is a chaperone in the lumen of endoplasmic reticulum which belongs to the category of HSP 70. It prevents the aggregation of protein. Basically the function of this BIP क्या होता है? This BIP, it binds to the newly synthesized polypeptide chain and do not allows their folding until the complete synthesization of polypeptide. As well as this BIP, it helps in the facilitation of this polypeptide, है ना. जो ट्रांसलेशन प्रोसेस है उसके थ्रू जो प्रोटीन सिंथेसाइजेशन हो रहा है उस पॉलीपेप्टाइड को एंडोप्लाज्मिक रेटिकुलम के ल्यूमन में ट्रांसपोर्टेशन के लिए भी जो BIP protein है, it also help in that particular process. So we can say that the chaperone, it is the chaperone in the lumen of endoplasmic reticulum. It prevents the aggregation, or we can say the binding, folding of protein before the complete synthesization of polypeptide. It recognizes the misfolded protein. जो misfolded protein है उसको recognize करता है and do not allows their transportation into the Golgi body. It helps the transition of protein into the lumen, जो अपन पहले ही बात कर चुके हैं. It facilitates the movement of polypeptide into the lumen of endoplasmic reticulum. So these are some function of this BIP chaperone. Now the next function is the disulfide bond formation. है ना? Disulfide bond formation, it also occurs within the oxidizing environment of rough endoplasmic reticulum. For this, there is a presence of another protein enzyme which is known as protein disulfide isomerase. Okay, there is a presence of protein disulfide isomerase which plays an important role in the formation of disulfide bond in these secretory protein. Suppose this secretory protein, if there is a presence of two cysteine. Now cysteine, it is basically an amino acid which contains sulfur in their side chain. अगर अपन स्ट्रक्चर की सिस्टीन की बात करें, and there is a structure which is like this. There is a presence of NH2, C, COH, H, and there is a presence of CH2, SH group in their side chain. So if there is a presence of two cysteine in front of each other under the oxidizing environment provided by the rough endoplasmic reticulum, and there is a presence of enzyme which is known as protein disulfide isomerase, then there is a formation of disulfide bond. Now, if we talk about the structure of this protein disulfide isomerase, it contains four subunits. There is A prime, B, and B prime subunits. While the catalytical subunits, these are A and the A prime, which contains two sulfahydryl group. Each contains two sulfahydryl groups like the protein glutathione. ठीक है, उसी के तरीके से फंक्शन करेगा. थोड़ी सी बात कर लेते हैं किस तरीके से प्रोसेस होता है. सो इफ देयर इज अ प्रेजेंस ऑफ ए पॉलीपेप्टाइड, if there is a presence of a polypeptide that contains two cysteine in front of each other, then there is a formation of a disulfide bond, and the enzymes which plays an important role in this process, it is known as protein disulfide isomerase. करेगा क्या ये जो H है, this PDI, it will take up these hydrogens, यहां से ये ले लेगा, sorry, these hydrogens, they will be taken up by the enzyme PDI, and then there is a formation of a disulfide bond between the two cysteine. Now these cysteine which remain bounded by the disulfide bond, they are called as the cysteine. इसे अपन अब कहते हैं, there is a cysteine, and these cysteines, they absorbs the UV radiation of 280 nm like the aromatic amino acid. So aromatic amino acid तो absorb करती हैं, but if the two cysteine they remain bound by the disulfide bond, these are known as cysteine and can absorb the UV radiation of 280 nm. So in this way, there is a formation of a disulfide bond within the lumen of endoplasmic reticulum. So इसको वापस अपन प्रोटीन को कहाँ डाल देते हैं? In the lumen of endoplasmic reticulum. So within lumen, there is a presence of a PDI which leads to the formation of a disulfide bond. Now, this after the formation of disulfide bond, this PDI it will get reduced. ये reduce हो गया है, तो अब इसको oxidize करना है. To oxidize this, we can say protein disulfide isomerase, we have another adaptive protein which is known as ERO1. In ERO1, there is again the presence of 2-sulfahydryl group which will take the hydrogens from the protein disulfide isomerase, and it will again become, or it again comes into the oxidized form for the formation of another disulfide bond. So in this way, there is a formation of a disulfide bond within the lumen of endoplasmic reticulum. So another function of rough endoplasmic reticulum is the formation of disulfide bond due to the oxidizing environment within the endoplasmic reticulum lumen, and there is a presence of a protein disulfide isomerase which contains the 2-sulfahydryl group that are involved in the formation of disulfide bond. These disulfide bonds, they provide the extra stability to the proteins. Now next function, if we talk, it is the N-linked glycosylation. Now the glycosylation is the process of addition of sugars onto the protein molecule. Now if the protein, it contains the consensus sequences in the form of asparagine X serine threonine. Asparagine X serine threonine. X is any amino acid except proline. Proline को छोड़ के कोई भी amino acid हो सकता है क्या? X. If there is a presence of these type of consensus sequences onto the protein, then there is a glycosylation, that is the addition of sugar onto the protein molecule. Now the sugars are generally added onto the asparagine amino acid. Suppose we are having the polypeptide which is containing the disulfide bond due to the activity of protein disulfide isomerase, and if it contains the consensus sequences in the form of asparagine X serine threonine. If we talk about the structure of asparagine, it is the amide of aspartic acid which contains the NH2 group in their side chain. So these NH and COH group, they remain in the formation of a peptide bond, and there is a free R chain that contains the CH2CO NH2 group. Now onto the membrane of rough endoplasmic reticulum, we have a derived lipid. There is a presence of a derived lipid which is known as dolicol. This dolicol, it provides the surface for the synthesization of some oligosaccharides. So it provides a surface, we can say, for the synthesization of oligosaccharides, And there is a presence of another enzyme which is known as glycosyl transferase, which transfers this oligosaccharides onto the NH2 group of asparagine amino acid, and the process is known as N-linked glycosylation. कैसे समझने की कोशिश करते हैं इसको? So we have N-linked glycosylation. For this, there is a presence of consensus sequences in the form of asparagine, serine, threonine. We have dolicol, on which there is a synthesization of a oligosaccharide. Oligosaccharide. Now this oligosaccharide, it is generally made up of 14 monomeric units. There is a presence of we can say 2N-acetyl glucosamine. There is a presence of, this is one N-acetyl glucosamine which remain attached to the dolicol. There is a presence of another N-acetyl glucosamine monomer, followed by 9 mannose monomer, followed by 3 glucose monomer. So total number of monomeric units, if we calculate, there is a presence of 3 plus 9, that is 12. There is presence of 2 N-acetyl glucosamine, and there is a total 14 monomeric sugars that together form a oligosaccharide on the dolicol, or we can say the dolicol. It provides the surface for the synthesization of 14 monomeric units, oligosaccharides, we can say. So this protein, if this contains the asparagine, serine, threonine consensus sequences, there is a presence of dolicol where there is a synthesization of we can say pre-processed or pre-constructed oligosaccharide in the form of 2 N-acetyl glucosamine, 9 mannose and 3 glucose, and there is a presence of an enzyme which is known as glycosyl transferase. Glycosyl transferase. Now the function of this glycosyl transferase is to transfer this oligosaccharide from the phosphate of dolicol onto the side chain of asparagine amino acid. There is an intramolecular condensation reaction which removes, we can say, the water, and then there is a formation of a bond between the sugar, that is N-acetyl glucosamine, and the asparagine of asparagine amino acid which remain present on the polypeptide. So if there are protein that contains this sequence, there is a glycosylation in the form of oligosaccharides, and these oligosaccharides, they are made up of 14 monomeric units that include N-acetyl, 2 units of N-acetyl glucosamine, 9 mannose and 3 glucose. The process is carried out by the enzyme glycosyl transferase, and this is called as the N-linked glycosylation. Here the sugars, they are added in the form of oligosaccharide. Sugars are added onto the N terminus of asparagine amino acids. That's why it is known as N-linked glycosylation. It is the N-linked glycosylation that occurs within the lumen of endoplasmic reticulum. So it is another function of endoplasmic reticulum. Now the next function, it is the protein folding and the processing. एक प्रोटीन के बारे में अपन पहले बात कर चुके हैं, that is the whip binding immunoglobulin protein, जिसको अपन ने कहा था it binds to the newly synthesized polypeptide chain. It keeps the polypeptide chain in the unfolded configuration. It helps in the protein folding. It helps in the facilitation of protein into the lumen of endoplasmic reticulum. Another proteins which remain present in the lumen of endoplasmic reticulum, these are calreticulin and the Calnexin. Now, if we talk about the calreticulin, it is the protein which remain present within the lumen of endoplasmic reticulum, while the Calnexin, it remain present on the membrane of endoplasmic reticulum. If we talk about the function of calreticulin, it is a calcium binding chaperone in the lumen which maintains the calcium homeostasis within the lumen of endoplasmic reticulum. It generally binds to the monoglucosylated N-linked glycoside. These are the N-linked we can say oligosaccharides, which attaches to the asparagine amino acids. Within the lumen, we are having the presence of another enzyme in the form of glucosidases, and these glucosidases generally cleaves these terminal glucose. And if there is a cleavage of two glucose and only one glucose left in this, we can say oligosaccharide, then this calreticulin, they can recognize this monoglucosylated and linked oligosaccharides. Then there is a binding of these, calreticulin, as well as the calnexin. The calnexin, it also binds to the monoglucosylated and linked oligosaccharide. Now, the function of these calreticulin and calnexin is to help in the process of protein folding. प्रोटीन फोल्डिंग के प्रोसेस के अंदर दोनों ही कैलरेटिकुलिन एंड कैलनेक्सिन ये हेल्प करते हैं. नाउ दे एंश्योर्स द प्रॉपर फोल्डिंग ऑफ दीज ग्लाइकोप्रोटीन्स. दीज आर द ग्लाइकोप्रोटीन्स, क्योंकि इस पे आपने पहले क्या ऐड किया है, शुगर्स को ऐड किया है इन द फॉर्म ऑफ ऑलिगोसैकेराइड्स, और उन्हीं ऑलिगोसैकेराइड्स को प्रोसेस होने के बाद में it is recognized by the calreticulin and calnexin, which helps in the process of protein folding. One, it remains present within the lumen of endoplasmic reticulum, while the calnexin, it is the membrane bound chaperone which remains present in the membrane of rough endoplasmic reticulum. Now this calnexin, it works with the calreticulin and ensures the proper folding of the ग्लाइकोप्रोटीन. सो जो आपके कैलरेटिकुलिन है या कैलनेक्सिन है, इनका काम होता है टू हेल्प इन द प्रोसेस ऑफ प्रोटीन फोल्डिंग. एक ल्यूमन में है, एक एंडोप्लाज्मिक रेटिकुलम की मेम्ब्रेन पे है. दोनों ही मोनोग्लूकोसाइलेटेड ऑलिगोसैकेराइड से ही बाइंड होंगे एंड हेल्प करेंगे इन द प्रोसेस ऑफ प्रोटीन फोल्डिंग. सो ये कुछ काम होते हैं. वी कैन से इट इज अ फंक्शन दैट इज द प्रोटीन फोल्डिंग एंड द प्रोसेसिंग. प्रोसेसिंग में भी हेल्प करते हैं और फोल्डिंग के अंदर भी हेल्प करती है. कौन? द एंडोप्लाज्मिक रेटिकुलम. सो द एंडोप्लाज्मिक रेटिकुलम, इट इज आल्सो इन्वॉल्व इन द प्रोसेस ऑफ प्रोटीन फोल्डिंग एंड द प्रोटीन प्रोसेसिंग. नेक्स्ट अगर अपन बात करें द फंक्शंस, एक फंक्शन रह गया, दैट इज द क्वालिटी कंट्रोल. सो the rough endoplasmic reticulum, it also controls the quality of protein synthesized within the lumen. Because if there is a misfolding of protein, if some of the protein they get misfolded or misglycosylated, उस condition में क्या होता है, these proteins they are sent, in that condition these proteins they are sent into the cytoplasm, and within the cytoplasm there is a addition of ubiquitin on these protein, and then these proteins are sent to the proteasomes for their degradation. आगे बात करेंगे. There are two sites for protein degradation. One is lysosome, another is proteasome. अगर polyubiquitin ऐड करते हैं प्रोटीन्स के ऊपर, the degradation it occurs within the proteasome. If there is addition of a monoubiquitin, the degradation of protein occurs within the lysosome. So if there is a misfolding of protein, then these proteins are sent into the cytoplasm where there is an addition of polyubiquitin, and the protein it will get degraded in the proteasome. So we can say that the rough endoplasmic reticulum, they are also involved in the process of quality control, that is the ERAD, that is the endoplasmic reticulum associated degradation of proteins. And this, in this process, these calreticulin, calnexin, they plays an important role for the, we can say in the process of protein degradation, or we can say in the process of misfolded protein degradation. So these are the functions of rough endoplasmic reticulum. Now we will move towards the smooth endoplasmic reticulum. If we talk about the smooth endoplasmic reticulum, the major function is lipid synthesis, जैसा अपन ने पहले बात की थी. All the glycerol phospholipids, है ना, all the glycerol phospholipid, चाहे वो phosphatidylcholine, choline, serine, ethanol, amine हो, they synthesized within the smooth endoplasmic reticulum. If we talk about the sphingolipids, where the framework alcohol, it is the sphingosine, है ना. यहाँ ग्लिसरॉल है, यहाँ पे फ्रेमवर्क अल्कोहल रहेगा sphingosine. In case of the sphingolipids, ceramides forms within the SER. It is very much important. जो सिरेमाइड है, जैसा अपन ने बात की थी, सिरेमाइड क्या है? इफ देयर इज अ प्रेजेंस ऑफ ए स्फिंगोसिन and there is a presence of a fatty acid chain, then the structure is known as ceramide. This ceramide structure, they again forms within the SER, but the formation of sphingomyelin कब बनेगा? when there is a addition of a phosphocholine, है ना, phosphocholine onto this molecule, on this ceramide. This ceramide, it is made up of sphingosine and the fatty acid. On this ceramide, if there is addition of a phosphocholine, it will lead to the formation of sphingomyelin. This sphingomyelin formation, it occurs within the Golgi body, जिसके बारे में आगे अपन बात करेंगे. Similarly, the sugars, the attachment of sugars that lead to the formation of, there is an attachment of sugar on this ceramide. It will lead to the formation of glycolipids, and these glycolipids again they synthesize within the Golgi body. So we can say that in case of smooth endoplasmic reticulum, there is a formation of all the glycerol phospholipids. There is a formation of a ceramide, but the synthesization of sphingomyelin and the glycolipids, it occurs within the lumen of Golgi body, जिसके बारे में आगे अपन बात करेंगे. Now again, in smooth endoplasmic reticulum, there is a synthesization of cholesterol, triglycerides, as well as the steroids, hormone like corticosteroids, mineralocorticoids, and estrogen, progesterone. These we can say steroidal hormones, they synthesized within the smooth endoplasmic reticulum. So it is the first function of smooth endoplasmic reticulum. If we move towards the second function, it is the carbohydrate metabolism. And the function is, they play an important role in the glycogen breakdown. During the process of, we can say, gluconeogenesis, जैसा कि अपन ने पढ़ा था in our biochemical pathway, during the process of gluconeogenesis, it is the complete reversal of a glycolysis except the three irreversible steps. तीन irreversible steps अपन ने discuss किए थे, and one of the irreversible step, it was the conversion of glucose 6-phosphate into the glucose. Now this conversion of glucose 6-phosphate into the glucose is carried out by the marker enzyme of smooth endoplasmic reticulum, or we can say the marker enzyme of smooth endoplasmic reticulum remain present within the liver cell. That is the glucose 6-phosphatase. ये glucose 6-phosphatase क्या करेगा? 6th पोजीशन से phosphate को remove कर देगा glucose के, and there is a conversion of glucose 6-phosphate into the glucose. So we can say the SER of liver cells, they plays an important role in the process of carbohydrate metabolism by reversing one of the three irreversible step of glycolysis, that is the conversion of glucose 6-phosphate into the glucose. Similarly, the another function of smooth endoplasmic reticulum is the calcium storage and release. As we all know that if the calcium it remains present within the cytoplasm, the muscle will remain in the contracted form. And if we want the muscles to relax, then we have to send the calcium back into the so we can say endoplasmic reticulum. So the calcium ion storage and release, we can say the smooth endoplasmic reticulum, they plays an important role in the process of calcium ion storage and the release. The muscles ER, it is known as SER, that is the sarcoplasmic endoplasmic reticulum, and the major function of these SER is to release and absorb the calcium. If there is a muscle contraction, they releases the calcium into the cytoplasm. If the muscles relax, they absorbs the calcium from the cytoplasm. अगर muscle contract कराना है, the smooth or we can say sarcoplasmic endoplasmic reticulum, that releases the calcium into the cytosol. If you want the muscle to relax, then the calcium it will be sequestered or absorbed by the endoplasmic reticulum. So in this way, we can say the muscles SER, they plays an important role in the muscle contraction and relaxation by the calcium storage and release. The most important function of smooth endoplasmic reticulum, it is the detoxification, that they have ability to convert the active drug into the inactive suitable form that can be excreted, with urine, we can say. है ना. तो अगर we can say it is non-soluble form, if the drug is it is in the non-soluble form, it is active, and if it is converted into the soluble form that can be excreted from our body, then it is called the process is called as the detoxification, है ना. So the smooth endoplasmic reticulum, they contains a marker enzyme which is known as cytochrome P450 monooxygenase. There is a presence of cytochrome P450 monooxygenase that utilizes the NADPH and oxygen to convert the active drug into the inactive suitable form that can be excreted from the body. Now this cytochrome P450, it is a heme containing monooxygenase, है ना. कैसे काम करता है बात करते हैं. इट जनरली रिमेन लोकेटेड इन द मेम्ब्रेन ऑफ एंडोप्लाज्मिक रेटिकुलम लाइक द calnexins, and suppose we are having a drug which is insoluble, it is active, there is a presence of in the presence of oxygen and the NADPH and H plus ion, this cytochrome P450 monooxygenase ये क्या करता है. It generally carries out the breakdown of this oxygen into the oxygen atom. One of the oxygen, it will be attached to the R, to the active drug, we can say, that leads the formation of the inactive drug. So there is a conversion of this active drug into the inactive drug by the addition of one oxygen atom, while the another oxygen atom, it is attached to the hydrogen ion. One H plus ion, it is donated by the NADPH, while as we know that NADPH, it is containing the 2 electron and 1 hydrogen. So one hydrogen from the NADPH, one from the, we can say, lumen of endoplasmic reticulum, smooth endoplasmic reticulum. It will lead to the formation of water molecule, जिसके क्या बन जाएगा water molecule, and there is a oxidation of this NADPH into the NADP. So in this way we can say the smooth endoplasmic reticulum, they are also involved in the process of detoxification by converting the active drug into the inactive suitable form that can be excreted from the body. So this is all about the structure and function of endoplasmic reticulum. Thank you very much.